Et puis même aller gaspiller dans le truc, moi je pense que c'est pas con. Ouais.
Oh, because you're one of the common people?
No, because I was jealous. I don't understand how do you get here, and I have to.
I'm like one of the secretaries, I bring my own tea.
No. They have like
They have own clothes.
low level, high level center.
Aussi on a une petite
Ah ouais?
Moi j'aimais pas son petit extrême hobby et miel.
Il est pas où?
Goûte-le.
Le meilleur.
Goûte-le, sinon on vous dira des nouvelles.
Le meilleur. Toutes les façons, la gamme de parfums. La gamme de parfums.
Si un jour ça passe un truc.
Ouais, mais toi t'as les trucs de base, t'as le menthe, t'as il y a ils font des amandes, des parfums, genre cannelle, pêche meringue.
Cinnamon vache.
Ouais, miel cannelle, incroyable.
Maintenant il reste que les ballons à la fin.
Ah.
Ça tu l'as oublié.
C'est que...
Do you want tea?
No.
Good.
No, I was just making sure. I was not making something.
בתשע ומשהו. You were missing, right?
אין הקלטה רגע.
שמונה חודשים.
דני, אין לך את ה...
יש פה משהו. זה? זה מקליט אוטומטי? צריך להפעיל את ההקלטה. אתה אומר שזה מגביר, זה אומר גם שזה מקליט?
כן, כי הדלק של הכל פתוחה. זה ממש טכנולוגי.
Okay. Do you remember anything from last week? Yeah. Is that mostly everyone? No, I'm asking if that was the number of people in class last week as well? Or should we wait a couple of minutes?
I think you can start.
Okay.
It's being recorded, so then we'll see if you can.
Okay. So, let's see, I think this was the last slide, but let's see if we can. So we were talking about the piriform cortex. Actually, let me go back to... I'll just say it. So in the olfactory, so what we've already said is that sensory neurons choose only one gene of the about 1,000, depending on the species, and express just that one gene of receptor protein. And we've talked about the glomeruli, which are the location where axons of many sensory neurons expressing the same gene converge in the olfactory bulb. And then mitral cells that read this information in the olfactory bulb only receive information from a single glomerulus. So there was this labeled line processing from sensory neurons through the olfactory bulb and then we moved on to olfactory cortex. And I'll just remind you that there are multiple regions that we call olfactory cortex, just for the sake of the name. But mostly we're going to talk about the piriform cortex, and I'll later talk a little bit about the AON. Piriform cortex is this region over here. I'm not going to discuss much, but let me just say now. So there's APC and PPC. A stands for anterior and P for posterior. Not clear whether there's really a boundary. So if you go from this side of piriform cortex to this side, there are some changes. Whether they're gradual or there's a boundary is not clear. The main change is the amount of input that is coming from the olfactory bulb is much higher here than here, and the amount of association inputs from other cortical areas is larger here than here. Okay, so you can think of it as like this is more primary and this is more secondary, but the boundary is not clear whether that even exists. Okay, did I mention what LOT is last week? No. So LOT is the lateral olfactory tract. So this is just the tract where the axons from the olfactory bulb go out of the bulb and into piriform. Okay. And I showed you the circuitry. There's a diversity of cell types, some inhibitory, some excitatory. The principal neurons are pyramidal neurons. So that's part of the reason why this is cortex. And I mentioned that there are less layers than what you're used to think about for sensory cortices. This is 3 layers. Layer 1, mostly axons. There are some GABAergic neurons. Layer 2 already has some pyramidal cells. Layer 3 is what we call deep pyramidal cells. Okay, these are superficial pyramidal cells. And then there's another layer that is not clear yet whether it's really another layer of piriform or something else. But this is just called the endopiriform. We're not going to discuss that. Okay, so this is where we were. And then the question was whether piriform cortex still maintains the labeled line processing. So is it still that this pyramidal neuron represents information from a single receptor gene, okay, or single receptor protein type? And we looked at this in 2 different ways. So looking at the anatomy, if you look at one mitral cell, will it just project to one region in piriform cortex? And this is what they've done here. And then the other way is if you look at just one region of piriform cortex or just one neuron in piriform cortex, where are all the inputs, the mitral cell inputs located? Okay, are they just nearby one glomerulus or are they spread over the bulb? And both these experiments, these projection analyses show that piriform does not maintain the labeled line. Okay, so this is, in this experiment, what they've done is label the mitral cells that belong to one glomerulus. Okay, this is typically, remember how many mitral cells per glomerulus? About 10. Yeah. So about 10 mitral cells, and then you look at their projections, and this is this yellow thing. So first of all, these 10 mitral cells don't just all project to one cortical region. Okay, you can see here AON, different parts of AON, anterior piriform, posterior piriform, lateral entorhinal cortex, olfactory tubercle. I actually forget what this is. But they'll also reach amygdala and some other regions. Okay. But even if you just look at piriform cortex, there are different locations in piriform cortex that are innervated by these single glomerulus outputs. Okay. And then the second question is, okay, so let's look at one piriform neuron. Where are its inputs? Do they all belong to one glomerulus? And this is an experiment where they've labeled using rabies from a single neuron in piriform, label all the inputs from the bulb. And so each color here, they're basically the green and the yellow are 2 different experiments, okay? Or 2 different parent neurons in piriform that they started with. So you can see that all the red is spread basically, sorry, this is a section through the olfactory bulb. So this is top, this is dorsal ventral, okay, top and bottom. And you can see that the red dots are all over. So they're not from just one glomerulus. The cells from one glomerulus would be something like spread over here, okay, this kind of region. And the yellow is just another example. But the important thing also is the green dots. The green dots are neurons that project to both these neurons they started from in the piriform. Okay, so you can also see that the same olfactory bulb mitral cell will project two different neurons in the piriform. So this is in line with what we've seen in the previous slide. Okay, this is just a different view from of the same thing.
So the difference between red and green is
So no, the red and yellow are two different starting neurons. Green is where you had both red and yellow. So neurons that are both projecting to both locations. Okay? So the labeled line is gone. And people have tried looking at what order is there. Okay, it's not labeled line, so what is it? So far, no one has found any type of order that you can describe. And the kind of, the models that people have in mind right now is that this is, to begin with at least, completely random. It's probably not, right? It's hard to think of something that is completely random. But what is the order, we don't know. There's a paper from Richard Axel and Larry Abbott, I think a week ago, in Neuron, trying to explain how totally random makes kind of no sense, because that means that the representation of an odor on your right hemisphere and left hemisphere is completely different. How do you even, so if you smell an odor with just one nostril, and then with the other nostril, how would you know it's the same odor? And people have done the experiment. You do know it's the same odor. So how does that work? So it's probably not completely random, but they now have a model trying to explain how with still random you can do some things that we thought you can't. Okay. So one of the questions is now, what does this mean? So does this, if this one piriform neuron receives input from all of these, you could think either that it just responds to many, many odors, right? Just has a way wider receptive field, if you want to call it that. Or that this piriform neuron is looking for combinations of receptor types, right? So a mitral cell just tells you that a receptor type was activated. And we've discussed, I don't know if you remember, but that most odors we think are encoded by combinations of receptor types, right? The fact that a specific receptor is activated is not enough to tell you which odor it is, because there's overlap in the different odors will activate overlapping groups of receptors. So if it's the combinatorics that matter, maybe this neuron really listens to neurons from different glomeruli, and when they're all active, it tells it, okay, this is banana, right? So this is, again, something I think still most people think this is kind of the case to at least some extent. That the different inputs represent something when they're active together. And to look at this, this is work by Ian Davison. He asked whether you can activate, a really simple question, how many glomeruli do you need to activate to get spikes from a piriform neuron? Okay, does it just respond to any of these glomeruli, or do you really need a combination? And he did this experiment. The main thing here is the comparison between the olfactory bulb and the cortex. Okay, so this here is the olfactory bulb. And if you record from a mitral cell, this is the map of the olfactory bulb. This is traces from a mitral cell. And these pink spots are spots of light that he used to activate the olfactory bulb. This is not channel rhodopsin. This is not optogenetics. But what they've done is glutamate uncaging. Is everyone familiar with this? Anyone not? Everyone. Okay. So I'll still say what it is. Glutamate uncaging, you have a compound that contains glutamate but is bound to some other compound. and this binding can be cleaved with light. So when you shine light, UV light, you get glutamate there, right? And then you activate synapses, and that's exactly what they've done. And you can see that this mitral cell responds to this one spot. So this is probably the region where the glomerulus is, right? That this mitral cell sends its dendrite into. And then if you activate more spots, different combinations, as long as you activate this spot, it doesn't matter what else happens, okay? Because mitral cells really mostly reflect just one glomerulus. Now you do the same thing in piriform cortex, and what they found is that almost none of their neurons could be activated with a single spot, right? So it's not you don't just try once, you play around with the location of light, but typically they just got nothing with 1 or with 2, and their kind of magic number was that typically 9 was the threshold. Now this doesn't mean 9 glomeruli. Why not? What is 9 here? 9 of what? Probably way more. It's 9 spots of light, right? So the question is now what you activate with a single spot of light. That would determine 9 of what. So the spots of light, you never know whether you're activating a single glomerulus, 3 glomeruli. So we don't really know the number of glomeruli. But it's probably more than 9 in their case, right? You're probably activating either 1 or more. Okay. So piriform cortex seems to be integrating inputs from multiple mitral cells, not to just be more active, but to kind of look at combinations of receptors. Okay, and this is how we think of as the codes for odors. So the mitral cell doesn't know which odor is present, but the cortex may already know. Yes. So the uncaging is done on the surface of the olfactory bulb, the dorsal surface. And the recordings are done here in the olfactory bulb and here in piriform. Okay, is this clear? Yeah. Okay. So there are not many experiments looking so far at coding of odors in the piriform, but one of them that is really heroic, again from Richard Axel's lab. Let's turn off the light. So what they've done is two-photon imaging in the piriform. And why is this heroic? It's mostly because of where piriform is, right? So piriform is somewhere here. So if you are used to thinking of people making cranial windows somewhere in the top, here you have the jaw. So the only way to get to see the piriform cortex is to remove the jaw of the mouse. And I forget how long they said the surgery took them, but it's a really long surgery. You cannot do anything awake ever. This is terminal experiments. So they labeled piriform neurons with GCAMP, and then they had this anesthetized mouse with the jaw removed and a window here so they can see the piriform cortex, and then they start presenting different odors. Okay, so what you have here are the red dots just show you areas that pass the threshold. Okay, these are responding neurons for different odors. And what you can see is that there's a scale bar here, right? This is 200 microns, so this is about 1 millimeter. So the neurons that respond to this is a single chemical. This is not even a complex object, right? This is just one molecule type. The neurons that respond to it are spread all over piriform cortex, really wide ranges. And if you take a different odor, it's spread over the same range. It's just maybe different neurons. And interestingly, what they found is that if you present different odors, you get overlapping representation. So you don't. It still seems like it's not that each neuron. You don't have a neuron now for each molecule, even though they combine information from the bulb, and you can have maybe groups of neurons representing individual smells now, it still seems like this kind of messy representation where many neurons tell you that pinene is now present, but some of them will also be active for octanol. Okay. And they're really the strongest claim they made is that the number of neurons activated seems to be not dependent almost on anything. So not whatever odor you present, you get about the same number of neurons at whatever concentration you present. Okay, and this in your paper is relevant because if you increase the concentration and the number of neurons activated doesn't change, what does that tell you? What has to play a role? Firing rate in terms of coding, but how physiologically do you get this? So let's think about the receptors, right? A receptor responds to different odors, but if you increase the concentration, you increase the chance that a receptor will be activated, right? So if you increase concentrations, you expect more receptors to be activated.
But they get saturated.
Right, but then you get another one that is not yet saturated. You'll get more activity in the nose if you increase concentration. Why do you not see it in piriform cortex? Maybe, but what do you need there? What type of? Yes, you need inhibition to play a really strong role in to keep bringing the activity to the same level, right? Because if you increase the input, you need to have some local inhibitory circuits that keep it down, right? And piriform has really important inhibition. It's one of the locations where if you block inhibition, what do you get? Hippocampus as well. If you block inhibition in hippocampus or piriform completely, what do you get? You get epilepsy. Inhibition is really important to keep activity at a certain level. This is not true for most other sensory cortices. But some areas are more prone to epilepsy, and inhibition plays a really strong role there, keeping activity at the right level.
And when you increase the concentration, you see more activation in the olfactory bulb?
Yes. Yes.
So inhibition is at the level of the mitral cells?
I'll get to the inhibition in a second. Okay? I'm talking about the cortex. No, inhibition level of cortex. Yeah.
The numbers of neurons that have the overlap in the molecules themselves seems like not that major if we compare it to the number of neurons that were actually activated.
Oh. So this is hard to answer because if these are anecdotes, right? You have the animal there, you have time to present maybe 10 odors in different concentrations, you're done. Whether these 10 odors represent the world very well is always a question. So we don't know. The answer is that we don't know. We can say there is overlap because we've seen it. Overlap depends on which odors you choose, but what the range of overlaps is, we just don't know.
Did they try to measure molecules that are more similar?
Not really.
Very rare.
I don't know how they chose their molecules. But as we discussed early last week, similar is something we don't really know how to define. So you can use their intuition, and I'll get to that maybe later, which sometimes works, right? You use your intuition, and then the output that comes, let's say overlap, makes sense. Then you think your intuition was correct. But actually defining similarities, we have no clue. Okay. So this is what we know about cortex, mostly for now, in terms of coding. So you get distributed activity. Their claim is about 10% of the neurons seem to be activated for any odor at any concentration. When you increase the concentration, if you'd get the same neurons activated at the same level, how would you know that the concentration changes? I mean, obviously we can say whether a smell is high concentration or low, right? So actually what happens is that for different concentrations you get different neurons, which is, again, a little bit weird, right? You expect the same neurons to maybe fire more, but this is not what you see. Okay. So another interesting feature of piriform cortex, this is just to remind you the circuitry a little bit, is to what extent they receive inputs from which region. And it seems like piriform cortex, even the anterior part that I've discussed that receives more mitral cell inputs, already shows kind of association cortical features. And what this really means, this is a nice way of just saying that a lot of what drives these neurons is not the input from the lower layer, but actually, either feedback from higher levels or parallel levels, right? So from other cortical regions. And what people have looked at here is the range to which, if I activate neurons in piriform cortex in one region, how long will it travel within piriform cortex? Okay. Typically in sensory cortices, let's actually start from the typical. No, let's start from the experiment. So what they've done here is express channel rhodopsin in a small spot of piriform cortex. And then these are many things to just show that it works. But the idea is that you then record at different distances from where you infected piriform. Okay?
Why did they want to change that?
Because they wanted to understand where this interesting inhibition comes from. So when you activate with different neurons or different concentrations or different odors, how does the cortex maintain this level of activity? And the idea was that you probably need some local interactions, and these could be either driven by excitation, long-range excitation that will then activate some inhibitory circuits, or just long-range inhibition. And this is kind of what they're asking. So what they've done is just record. So this is where they infected, and then you record, further and further away. And this is basically the result here. You can record pretty far away, right? 2 millimeters away, and you still see very strong excitatory currents. And then they looked at the space constant of this. How fast does it decay with space? Okay, and the space constant they see in piriform is about 3 millimeters. If you compare that to S1 or V1, which is about 100 microns. Okay, so this is very atypical of primary sensory cortex. Primary sensory cortex neurons typically are mostly activated by their incoming input, from thalamic inputs, right, from sensory inputs. And these neurons seem to be very, very strongly activated by their neighbors, even long-distance neighbors. Okay, so this doesn't explain inhibition yet. This is just excitation. But they've done the same with inhibition, and what they find is that inhibition actually drops pretty quickly. So we're just going to look at this graph, okay? So now they're looking at iPSCs. Earlier they were looking at ePSCs. Now if you just don't block anything, so you just do the same experiment and look for ePSCs, you can see very long-range connections. But these could be either monosynaptic, so the neurons that you activated with light, some of them may be inhibitory and they have long axons. Or you can just be activating excitatory neurons, and these will activate local inhibitory interneurons somewhere far, and this is what they found. So if you basically block glutamate transmission now, so you're only left with a local interneuron activation, then it drops much faster. Okay, and it drops pretty much as fast. How fast it drops depends on the spot of channel rhodopsin. So direct activation by channel, just by light, without any synapse, is about the same width. So it seems like inhibition is really, really local, but long-range excitation can drive this inhibition even far away. Okay. So this is kind of atypical connectivity for primary sensory cortex. Okay. So I'm basically going to sum up. This was kind of an overview of what we know about olfaction. Obviously, there are more details, but this is the basic. And the picture that kind of emerges is this. You have the olfactory epithelium. Each line ending here is a sensory neuron. You have sensory neurons expressing different receptors, not just 3. Their axons converge in the olfactory bulb. So information from receptor types converges in the olfactory bulb to form glomeruli and glomerulus-specific mitral cells. And then input over from multiple receptors converges in different combinations in the olfactory cortex. Okay? Yeah.
Just a question about the last slide. So what do you mean
This
by...
one?
Yeah. I guess looking at the scales, but by like long distance, what is long distance?
Long distance. So let's go one slide. So in pure form, what they find is that the lambda, how fast these decay, right, is about 2 millimeters. And this is compared to S1 and V1, which is about 0.1 millimeter. Okay. Okay. So basically, if you now want to understand how the identity of an odor is encoded in these different regions, you have here the combinatorics of different receptors. Right? Here, still labeled lines, so still combinatorics of receptors. You add some, in both of these, you can add some temporal aspects that we've discussed, the relationship between the time of firing and the phase of sniffing. And you have here pure form that now combines different receptors and produces different output patterns, right? What else do you want to know? This tells us everything, right? We now know how each odor is encoded in each region. Are we happy?
We also remember.
Yeah, so this projects to some memory regions in hippocampus, and you can go study hippocampus. But what else do you want to know? Yeah. Yes. Good. What else? So basically what I've showed you here is that if your goal in life is to discriminate between 2 odors, you can do the job, right? You read this input, read this input, read this input. You can discriminate between 2 odors, right? Each one of them, I forget. Yeah. You get unique responses to each odor, and you can discriminate whatever you want. But is this really what a sensory system needs to do? Just discriminate between stimuli? Probably not. So now, let's talk about what questions are completely unanswered. And I'm going to just describe some attempts to answer them because they're still not answered. But I think this is where things become interesting. So far, this is like, okay, where different odors are, where are the codes different? But that's really one bit of information. So what other questions I think are important? So these are 3. These are not all other questions. These are just 3 that I can describe some attempts that are going on now in different labs. So this is the one you mentioned. Where is the odor? I mean, it's great that I recognize that there's the smell of food. So as humans, if you recognize the smell of food, you know where the kitchen is. You just go there. And probably other animals can use similar information. But often many animals need to use the sense of smell to get some sense of where things are as well and find them. So how do they do that? Another one is concentration and variance. So if we, as we said, responses in pure form and in any other region are going to be different when you increase concentrations. So how come you know it's the same odor? Okay? In a second, I'll tell you why I think these are all difficult problems, where the difficulty stems. And the last one, which I'm going to talk about the most, because this is what I do, is background invariance. So it's great that you can identify an odor when it's the only thing in the world, but this is never the case, right? You'll never smell food as the only smell around. So how do you segregate the smells of what you care about from the smells of the background? So why are these difficult? Let's go over them. So concentration and variance. I'm not going to show you a paper, but I'm going to explain why I think it's a difficult task. Okay? Yeah.
I know this wasn't in your list, but do we know how we recognize smells that we've never smelled before as good or bad or something we like?
Some people think they know. So I would say no. But I described in the beginning of last week, there was this study that used PCA to look at the perception space, right? So what they did is they took these lists of, flowery, chocolatey, whatever, poop, anything you can think of, right? And you just rated different odors. And then they took many of these ratings and they did PCA and they found an axis that represents perception the best. They claim pleasantness is that axis. Okay, so maybe that's an explanation. So what you do is you look at which of the original axes have a long projection on that axis. So if you see all the flowery, chocolatey on that side and all the poop, whatever, on the other side, then you can say pleasantness. It's not really well defined. Yeah. Yeah, so I would say really no. We don't know. Okay. So concentration and variance. Why is this a difficult problem? So let's look at this graph here. Okay, this is odor concentration on the x-axis and the response on the y-axis, and each color is one receptor type. Okay, one gene. So let's take the blue. The blue responds to this odor, whatever it is. If you increase the odor, it responds more, and then it saturates, right? And then you can take the red. It basically does the same thing, just requires higher concentrations, okay? So if we now take this odor at this concentration, where this dot is, it's encoded. Let's say these are the only two receptors in the world right now, okay? So this odor will be encoded by the activation of the blue receptor, right? But when you increase the concentration over here, now you get the input of both receptors active. So the combinatorics, the input that you receive, has already changed. Just assuming that banana is always the same receptors that are going to be activated, it just doesn't work, because you increase the concentration, you all of a sudden get more receptors of different types. Okay? Is this clear? So it's nice that we think that each odor is encoded by a combination, but you're going to get a different combination for different concentrations. So this requires some explanation.
If I understood, you said that in the peripheral cortex, for different concentrations, I will have different neurons that will be activated.
Yes.
So are there neurons that are more specific to low concentration and neurons that are specific?
No one has done that experiment. We don't know. So the imaging data that I showed you is it's almost an anecdote. They have done this experiment once with a bunch of odors and a few concentrations, and it just doesn't behave simply. So that's the main take home. There is no clear model of how it behaves. We just know that it's not simple. Okay?
And on the perceptual level, can we distinguish between the concentrations?
Yes.
Taste and smell?
Yes. So typically, yes. So this is, again, hard to answer in a really general way, because you'll need to try all odors in the world. And there are examples that work really weirdly. I forget which molecule it is, but there's one molecule that, at least at low concentrations, smells really nice, and then at high concentrations smells like poop. But that seems to be the oddball. Typically, you'll say it's the same odor, and you'll even rate it, with similar pleasantness. And you can tell, obviously, if it's high or low, which one is higher concentration, if I give you two. And if you think about animals that require the sense of smell for their survival, it can't be otherwise, right? If they don't know that food is food when the concentration is high or when it's low, then they'll die.
But it seems like we know it's the same smell, but we experience it differently when it's in different concentrations. Like when I think about examples of strong smell and not so strong smell, I feel differently about it. I know
In terms of how pleasant it is for you?
maybe my experience of the smell is different, though I know it's in the same source.
I'm not sure what you mean by experience of smell.
Maybe it's the pleasantness. I don't know.
Could be.
Yeah.
So what you're saying, maybe we don't need to be completely concentration invariant. Maybe it's just a range, and then outside this range something else happens. Could be true. But you need some concentration invariance, right? And this is I just want to explain why it's not a trivial problem, right? Because initially you imagine, okay, same neurons activated more, that's concentration, right? And I can say it's the same odor, it's just stronger. But this is not the case, right? So we need something else. And these are glomerular images, okay? So what you see here, these spots, this is the olfactory bulb surface. And these are glomeruli that are activated by either this odor or this odor, and then different concentrations. And what you can see for these two odors is, ethyl valerate and allyl butyrate are way more similar to each other at this concentration than allyl butyrate to lower concentration of allyl butyrate. Okay? So this is not a trivial problem. One second. And I'm not going to describe how it may be solved, even though I have the slides. I'm going to go through them quickly without anyone seeing, because that's the paper that they're going to discuss next week. Yeah?
You said that for different concentrations we have the same general activity in the...
In pure form.
Yes.
Yes.
But we do see differences in the olfactory bulb.
Yes.
And then it dissolves on the way to the...
It doesn't dissolve. We think it involves inhibition, the strong inhibition in pure form. So this is now has been shown in pretty much all cortices, I think, that there's active normalization of the input. So if you increase the input, the output is not going to be just way higher. And it's done by inhibition. So it receives a lot of input, but this input also goes into the inhibitor neurons, and they're going to bring everything down to the same level. Okay? Okay, so if we all agree that this is not a trivial problem, I'll move on. Do I need to give you a break? I forget. You want a break in the middle, or? Yeah? Okay, so let me go through one more problem, and then I'll give you a break. So, yeah? You didn't see this, right? Where is the odor? So I'm mentioning Noam Sobel too often in this class, but this is really nice work that he's done a long time ago. His claim to fame is basically showing two things. One is this PCA of perceptual space and chemical space, and the other is basically claims that humans are as olfactory animals as any other, which is just wrong. But what he does show is that we can do, and we're affected by smell more than we think, okay, which is, I think, very important. So we all know that, a dog could track, smells of things, right? So this pheasant was dragged here on this field, and then they put these lights on the dog and just and made a video of him sniffing through the field until he finds the pheasant. So what Noam did is, I forget the trait, I think it was chocolate or something more smelly maybe, and he had these Berkeley undergrads cover their eyes and just sniff the grass until they, and let them follow the trail. The point was that they could do it, okay? So this is one type of odor source localization, but this is a very specific type. Why is this a very specific type? Because if you take the odor and you smear it on the ground, then you make it stable. Okay, you have a path that you can try to follow. But what happens if the odor is just in the air? What do you do then? What would you do? How do you find the highest concentration? Gradient, right? That's what in biology is used a lot. the assumption that the gradient gives you the information of where, the mathematical gradient will give you the information of where the source is. Now, if you've ever, does anyone here smoke? Good. Has anyone seen anyone smoke? If you look at this cigarette here, this is the source, okay? This is very close to the source. This is far from the source. Where is the concentration higher? In this case, the concentration here is as high as close to the source. But if you go a little bit out, outside of these plumes, then you see nothing. So there's very little information in the gradient here just because of turbulence, right? And this is the typical case in air. This is not a specific case. So if you're smelling something in the air, following it based on these eddies, we call these increased concentration regions. There are different ways of seeing this, right? This is an image. This was actually done in turbulent water. This is measuring in a single location in time. So what you do is you release an odor from some valve, and you have PID, whatever other device you want to measure the concentration of the odor at just one location over time. And you get these peaks, right? Because these things are dynamic. So even if you start mapping this thing, in another second, it's different. So not trivial, okay? And people are working on this now, and most of the ideas are not really sensory in the way we think. It's really behavioral strategies, right? If I smell something here, where do I go next to smell? And there are two different flavors for this. One is, at any one point, I have some estimation of where the source is, and I go towards that direction, and then sample again. The other is, at any one point, I can estimate where I should move to get the best information. Okay? And that doesn't necessarily need to be towards the source. Not going to talk about these. So after the break, we're going to talk about background invariance. Okay.
מה המשמעות בעצם של המרחק הזה, הסטטוטורי? שהאם שהוא שונה באופן מובהק מהפקטורים... ומה זה היה? האודיטוריה שלו?
S1, סומטוסנסורית. התשובה היחידה שאני יודע להגיד זה דרך האיניביציה. שזה גורם לאיניביציה להיות long range, אבל יותר מזה אנחנו לא יודעים. עקרונית, יש לזה עוד משמעות, אבל זה נפנופי ידיים. אם אתה מסתכל על סנסורי קורטקס, זה יותר נפנופי ידיים בדרך כלל. כשמסתכלים בסנסורי קורטיסיס שונים, אז אם אתה הולך במעלה ההיררכיה, נגיד שאתה יודע מה ההיררכיה, כי גם זה לא טריוויאלי, אז יש יותר long range connections כאלה, לוקליים, ופחות השפעה של האינפוט שבא מהתחנה שמתחת. זה בפריפורם, כן. אבל זה קצת... המחשבה היא שאולי...
זה ל-V1 אלא ל-V...
כן, אז אולי פיריפורם הוא לא אקוויוולנטי, כי כל המערכת הזאת היא יותר רדודה. אז אולי זה... אבל המסר הוא באמת שלא לחשוב עליו כעל V1.
כאילו הבלט זה יותר V1, לא?
לא, עשיתי כבר...
אני לא יודע באיזה מובן הוא V1, אז...
בסדר.
אנשים אומרים, יש אנשים שאומרים שהבלב הוא המקביל לתעלמוס, אבל...
עכשיו זה להציג את שלה, החל
אני
פספסתי שיעור הקודם.
אני חושב שזה
אבל,
קצת... לא הרגשתי אי פעם שהאנלוגיות האלה עוזרות באופן מהותי. אבל זה להגיד שהוא...
תראה, זה משמעותי להגיד 200 נקרא לעומת 0.1 זה...
לא, התוצאה הזאת היא משמעותית. אני אומר להכריז שהבלב הוא תלמוס או V1 לא עוזר לי.
רק זה.
אבל אם אתה... מה אנחנו בדרך כלל חושבים על זה, אני חושב? למה יש לך V1 ואז V2 וזה? כי אתה צריך, קודם כל בשביל בכלל להתעסק עם התמונה, אתה צריך לפרק אותה להמון גורמים, וזה דורש את השכבות האלה. ובאיזשהו שלב אתה רוצה להכניס משמעויות לתמונה, כי פעם האחרונה שראית את האיש הזה חטף את הבוקס וכל מיני דברים כאלה, וזה לא יבוא מהעין, אלא עם הבוקס מהעין. אז זה ה-association fibers, הם נותנים לך אפשרות לחבר את האינפוט הזה לאיזשהו משמעויות. זה המודל. אז מה שאנשים אומרים, במערכת הריח זה מתחבר מאוד מוקדם אולי למשמעות. אבל...
זה לא פייר, כי הוא באמת מגיע אוקיי, זה לא חוכמה.
Me too. Just fix the footer. There's no where's the graphic?
הוא לוקח את הקורס הזה למקום מאוד מאוד נקרא לזה טיפולי מעשי כזה. אשכרה יום אחד אני בא לשיעור ופשוט כל השיעור היה שהוא הביא איזה מטופל שלו שישב ועשינו כאילו מין פורום
forgot that Mati asked me to tell you that Yoram will be next week. Because Yoram Ben Shaul, who's going to speak on the vomeronasal system, he was planned to speak the second hour today. But he's going to be next week. And then there's going to be one paper. Okay. So, background and variance. So if you imagine this mouse, interested in finding cheese, and he walks around outside, he may find this pile here. He's going to smell the combination of everything around, right? So how can he recognize the cheese? And again, why this may be a difficult problem. So let's think of the cheese as this specific glomerular pattern, okay? This represents the olfactory bulb, and these are glomeruli, and these three glomeruli, when they're activated, the mouse knows cheese. And he walks around here and he gets this input, right? Because there's cheese plus some other things. So he may think cheese is there and find it. But you can think of these three being activated in this case because there's banana, which is the green ones, and fish, which is the blue ones. Okay, so how would he be able to tell these two situations apart? Now, again, I'm not going to give you an answer because these are the unanswered questions for now. But the way that I think about this is that this is an olfactory equivalent of the cocktail party problem. Have you all heard about the cocktail party problem? Yeah. So why do I think this is equivalent? Because you're interested in only part of the scene around you, right? You get a mixture of inputs, but you're not interested in everything. And what you are interested in may change very quickly. This is a dynamic thing, right? Now you're hungry and you want to smell the cheese, and then you think you sense danger and maybe you want to smell the cat. So this will change very quickly. And changing very quickly, what I make of this means that it's not that you learn. This is not going to be a learning process that you stand here for now many minutes and synaptic plasticity is going to allow you to increase responses to cheese and maybe decrease responses to other things. You change quickly, right? If I become boring and you want to read something on the internet, that's not going to take you hours to do. So, and then there's one third thing, and that is that the different inputs that are competing, I'll say it, competing maybe for your attention, are already activating overlapping groups of sensory neurons. So the interaction between the different objects is already at the level of sensory neurons. Why is this? Because different people speaking, they use pretty much the same range of frequencies. That doesn't, you can still listen to one person and not the others. And also different objects here may activate overlapping glomeruli, right? Overlapping receptor types. Okay. So, yeah.
How is that behavior in cortex level different from when we change the concentration?
We really don't know. So there is no data whatsoever about the representation of an odor and a background in the cortex yet. But if you want to join my lab, you can do it. Okay, so yeah, and when I think about how this system works, I'm assuming that there's most people I think assume that there is some system more internal, right, more central in the brain than the sensory regions, that somehow makes a decision of what is important, who is the person I should listen to, and then uses this information to influence the sensory regions. So A1, and people have done this, A1 is going to respond differently to a stimulus if it matters to some reward or if it doesn't, right? And this is probably done by some top-down inputs that tell it, this stimulus is really important. So this is also how I think about this system, and I already mentioned this. but we haven't discussed any physiology related to it. That olfactory bulb projects to these cortical regions and others. There's about 10 times more axons going back from piriform cortex and AON compared to the feed-forward direction. Okay, so you could think, what I'm thinking, that maybe these axons convey information of what is important right now, and use this information to shape the responses in the olfactory bulb to make sure that the cheese is processed adequately and the fish is lost in some background. Okay? Is that a question?
But I thought can you show that, like the model you showed with the banana and, it's glomeruli. Okay. Okay, and I thought it's in the cortex level.
No, this is
Okay.
input. This is the nose, really. Okay. So this is basically the model that I'm proposing, and I'm going to show you what we've done so far in trying to figure out whether this works or not. So the first thing just to mention that these inputs have been probably discovered by Monica Chal. So we know that these inputs exist for over 100 years, but for a combination of different reasons, the physiology is not really known, and they haven't really been studied deeply. So this requires really different levels of investigation. You want to know the physiology of these circuits, and you want to know something about the behavior, and then you want to try to link them. So now I'm going to show you some physiology and some behavior, and we haven't linked them yet. But the first thing we looked at is really what is this physiology. So what kind of modulation can these inputs perform in the olfactory bulb? Okay, and the way we approached this was, first of all, just let's activate these axons in an anesthetized animal or slice. Well, we did both, and see which cell types receive this information. Does it inhibit output, excite output, and how does this depend on different things? So these green axons, they're green because they're expressing GFP. The other colors are just to code the cell types. So these are axons coming from olfactory cortex, and I didn't tell you, but we specifically labeled the AON for no specific reason other than knowing that a different group was doing piriform. And everything that I'm going to tell you, we didn't do the experiment in the exact same way. So some things are a little bit hard to compare, but most things seem to be very similar for both of these. Okay. So we labeled them with GFP and channel rhodopsin, and now we can activate them with light. Why is it important to do this with channel rhodopsin and not just electrically stimulate?
So it's more specific.
Yeah, so really I think the best contribution of optogenetics so far is for in vitro experiments, even though it's really helpful for publishing high-profile behavior papers. You read them and then you're not sure what really happened. But really what it allowed to do is, you have here these feed-forward axons and these feedback axons. And if you put an electrode here and just apply current, you may activate both. So what channel rhodopsin allowed is I'm going to go back one more, is to separate basically what you do is you inject your virus here, but then you shine the light here, right? So only axons that are in both of these regions will be activated. Okay. So first thing that we looked at is really to which layers in the olfactory bulb these axons reach. And what you can see is that in the deep layers, granule cell layer, you see a lot of them. And then there's not much here, and then you see them again in the glomerular layer. Now this is kind of surprising. How do they reach here without passing here? The reason you don't see them here is because what we see is mostly the endings of these axons. Okay, so it's not entirely, it's not purely the synapses, the presynaptic boutons, but they're really overexpressed in this. So this was known. This is really what Monica Chal already said, that granule cells seem to be the ones receiving the feedback. He didn't know where the feedback comes from, I think he mentioned cortex as a possibility, and he didn't know whether it's excitatory or inhibitory, but he could see that they're going into the granule cell layer. So they seem to be going really into all cellular layers. There are some neurons here, but most neurons are either mitral cells are down here, granule cells are deeper, and then all these periglomerular neurons around here, all layers. And I'll cut the story short. Basically what we've done is record from each cell type in slice. So in slice you can really target which cell type you want to record from, and then activate these axons and ask who is activated. And the answer is that all of them. Okay, so it doesn't just reach all layers. Any cell type that we could identify receives direct input from AON, but not all to the same extent. So when we looked at the effect in vivo, so this is now, is everyone familiar with these plots? Yeah. Okay, so each tick here is an action potential. This is one trial, another trial, 5 trials here. Blue square tells you when the light was on. Okay, so this is the time when these axons are activated. It's actually 2 smaller bars. It's 2 stimuli, but it doesn't matter. And then, so what you can see here is that these neurons, mitral cells, I already mentioned this, they tend to fire in these bursts, which are related to what? In vivo? Yes, to the breathing of the animal, right? So whenever the animal inhales, you get a burst. And then you turn on the light, so it seems like here, if you look in these 5 trials, you don't see spikes, but 5 trials don't tell you much in a cell that likes to pause anyway. So you can take many more trials and make a PSTH, and you can see that turning on the light completely silences these neurons. Okay. But what we've seen in the in vitro case, in the slice, is that these also receive direct excitation. It was really weak, but we could see it. So, and we never saw it in vivo, and it took us, I think, took me something like 3 months to figure out why we don't see it. And it's just the size, the time slices that we chose. So these are, I think, 50 milliseconds. So you're averaging the really small excitation with the inhibition that follows it, and it's lost. But if you use 1 millisecond bins, which people don't do for PSTHs typically, there's no reason often, then you see this really 1 bar of excitation. So it's just a single action potential that increases at a specific time, increases its probability at a specific time, and then it's followed by inhibition. Okay? So what we learned from these experiments is that the major effect on the output of the olfactory bulb, the effect of activating cortex on the output of the olfactory bulb, is inhibition. But there's also this possibility of excitation. Now, in vitro, we then went and we tried to figure out when this excitation will actually generate an action potential. And what we found, I think I didn't put the slide here. Yeah, so let me draw it. So we found that if you just leave the cell alone at rest, okay, so you do a current clamp experiment. Everyone knows what that means? No. Current clamp experiments means that you just measure the membrane potential and you don't affect it. Okay? So the cell is at its own resting potential, and then you activate the cortex and you see this small excitation, but it should be this way in current clamp, and then you don't see action potentials. Okay? And then if you now make the cell fire, okay, so you inject current, so you depolarize the neuron, and now it fires a lot, and then you activate cortex, you're not going to add a single action potential. But if you do something in between where the cell is a little bit firing, but very low rate, and then you can add an action potential. Okay, so basically, this is not extremely surprising because if you've ever seen an FI curve, which is the frequency of firing as a function of the input, then you typically in a neuron get something like this, right? At some point, some current is just not enough, you get nothing, and then you get the threshold, you get more and more spikes until you get saturation. Each cell has its maximum firing rate, right? So your really small current is not going to be effective here, is not going to be effective here, it's mostly effective here, right? Okay. And I'll say in a second why I think this is important. Okay, so inhibition, now going back to our model of how this helps the mouse find the cheese. So let's say, now we activated all axons non-discriminatively, but when the mouse is activating them from within, he may be inhibiting the ones that are inhibiting regions that respond to background odors, and maybe using this excitation to get a little bit more firing from the cheese, right? From the neurons that respond to cheese. This is hand waving, right? It's just a possibility that we can now go check. Now, why is it important that this is not massive excitation? What would happen in this kind of model if your excitation is really strong and you're searching for cheese? So every time you're searching for cheese, you get all the mitral cells that respond to cheese to really fire. You hallucinate, right? You imagine cheese. So you don't want this excitation, top-down excitation, to be really strong for this kind of thing. Okay, so this is the physiology. We now know something about, there's some more details that I'm going to spare you. But we know that really the effect is strong inhibition and potential smaller excitation. Okay. Yeah?
If this model is true, there has to be something that integrates what we're looking for.
Yeah, of course.
So what is it?
So, I don't know. So somewhere, the regions that make the decision about where to focus attention have been studied somewhat in vision. I don't think we have an answer there either. But no one has studied that for any other sense, really.
Can you explain again what's the difference between the two?
This and this?
Yes.
It's the same data.
Yeah.
Just when you make the PSTH, how wide is your bar? So basically, this bar here, the first bar following light stimulation is averaging this whole thing. And then you take this excitation, average it with a lot of inhibition, it looks like inhibition. Okay. But I've never seen anyone make a PSTH for 1 millisecond. It almost sounds stupid, because the whole idea of PSTH is that you can't make sense of each millisecond. So we really had to get a lot of repetitions to see it, but that's details. Okay. So we have a model, right? Some sort of model, not what a physicist would call a model, but some hypothesis of how this works. So now the question is, do mice really find cheese in this kind of condition? Can they actually do it? Oh, too much back. How do I know that the mouse could really find the cheese? Maybe they can't. Maybe they just walk around until they get this really high concentration. So the next step was to start doing the behavior. And I think for a sensory physiologist or anyone studying a sensory system, this is the goal when you study behavior. To be able to describe behavior using a psychophysical curve that relates some behavioral ability, right, that whatever you're interested in, to parameters of the stimulus. If you can do that, you think you understand, right? So if I would now ask you to just detect this line, and I start playing around with the light intensity or the contrast of the image, I can predict what you're going to do, right? Whether you'll be able or not to perform this task. So this is what I want to do now with target odors and background odors. I want to know when. I want to be able to predict when a mouse is going to be able to find the cheese and when not. So how do we do this? And really the main difficulty here is we can train mice to do tasks by now, which is something new to neuroscience somewhat, right? People used to think that if you want to do behavior, either use humans, or if you need to record, okay, take a monkey, right? But about, 15 years ago, I think, people started training rats and mice to do really psychophysics. And it was, we can do it, so why not? But really, so this is not the difficulty. But the difficulty here is defining stimulus parameters, because as we discussed, we don't know how to define similarity, right? So contrast, in a way, is a similarity between your target and the background. So what is the contrast of a target odor and its background? How do I define their relationship? So initially, I actually didn't think about it too much. I just said, let's get some behavior working. And the idea was to train a mouse, present a puff of some odor mixture, but train the mouse to just tell me whether a specific target odor is part of this mixture or not. Okay? So this will just tell me if the mouse can detect the cheese or not. And this is kind of how it worked. So the world for these mice was 16 different odorants. So from now on, each square just is a molecule type. Okay, these are the molecules. I'm not going to tell you what they are for now. And then in each trial, so for each mouse we actually had 2 targets, and this is going to be important a little bit later. So 2 of these are targets, and this means if you detect them, you get a reward, right? The other 14 are what we call distractors. Whether they are there or not, whether you detect them or not, doesn't matter. You're not going to get anything for it. And then every trial, we present some random combination of these odors. So these are binary mixtures in the sense that each odor is a binary variable. Okay, it could either be on or off. We just present a random mixture and ask the mouse to let us know whether he's detecting the target.
The same concentration?
So we don't play with the concentrations. They were all in the same, in some way in the same concentration, but they end up not being the same concentration. But that actually doesn't matter because we and the mice have different sensitivities for different odorants. So the real best way to do this would be to find the threshold of the mouse for each odor, and then use some multiple of that. This would take you 10 years. Really, our main problem is that the best detector for any odor that we have in hand is the mouse. So when we detect with our machines that there's nothing there, the mouse could still report. Finding their threshold is a huge pain. And so we ignored that problem, and we thought, okay, one mouse will have these 2 targets, a different mouse will have these 2, a different mouse these 2, and this way we kind of average out the differences between the odors.
The size of the mixture was the same, like 6 odors?
No, so any combination from 1 to 14 is allowed. I'll show you in a second. Basically, this is the task, okay? Any mixture, if one of these 2 is on, I didn't tell you how the mouse reports. They lick. So they have the odor port, and then there's the water port. These are thirsty mice. So whenever they detect one of these 2 target odors, they touch this water port with their tongue. This closes a circuit. We detect that, and then water droplet comes out. Then they can also lick more and get that. But just to make clear, this is not classical conditioning. It's not that once the target is present, the water is out. They need to first report. Okay? Yeah. So how do we train them? So we started with the distribution of different mixtures, the number of components in the mixture. We start with this kind of distribution where really, 14 is allowed, but the chance of getting 14 is practically 0. You mostly just get single odors and sometimes 2 or 3. As the mouse reaches some criterion, typically 80%, we don't jump. We have 2 other distributions in the middle, but this is the data I'm going to show you is from this kind of distribution. Okay, so chance of getting a mixture of 2 or 14 or 6 is the same. Okay. This is the real nice thing. I used to say this a little bit more with confidence. I'll tell you in a second why I lost a little bit confidence in this. But going back to monkeys versus mice with behavior, is anyone here working in a monkey lab before I? No. So training monkeys, how long does that take? To do, do eye movements, whatever, move a manipulandum. Does anyone have months? Typically months, so 6 months or so. This is about 1,000 trials, which is about 3 days. Okay, so it's really quick. And for two reasons that I've learned part of them later. But one of them is that olfactory cues for mice are amazing. If you try to do the same thing with a visual stimulus, much simpler visual stimulus, let's say blue light on the left versus green light on the right, it takes about 2 to 3 weeks for a mouse to get that. Right? So this was really a fun part. You very quickly get to the point where you start gathering data. Okay, so we do this. We did this with 13 mice then, and this is what it looks like. So their head restrained during the task. Whenever there's this green circle here, it tells you that a mixture is being presented, and if it's green, it includes the target odor, and if it's red, it doesn't. And this is a well-trained mouse. This is the odor port. This is the water port. Oh, in this case, actually, we didn't close a circuit. There's an IR beam that the tongue breaks, but it doesn't matter. And one interesting thing is that all mice, when they know they're about to get a reward, they start running. Okay. So can they do it? Well, very well. So with these, the way we set up the task, we actually barely challenged them. So for a single, what we do here is now take all these trials and then just sort them by the number of components that went into the mixture and see how well mice did. For a single component, it's about 95%. For 14 components, it's about 85%, or what is it? I think it's 88%. So really, we're not close to challenging them. It turns out it's not as good because what they do, they have a very strong bias towards licking. So the Go trials, they get for free. Basically, to not lick, they need to be absolutely sure that the target odor is not there. So we ended up analyzing only the no-go trials, so the ones that do trick them, and try to find out what makes them fail. And there are two ways to look at this. So really the idea is that obviously the number of components makes them fail. You increase the number of components, but we don't think that all 10-component mixtures are equally difficult. There's something about the relationship of the specific molecules. So how do we describe that? Since I already told you that we don't know how to do that. So the first way that we looked at this was based on intuition. And as I said, you just try your intuition and see if the result makes sense. And the intuition was that we have a completely intuitive measure of similarity, and that is we introduced to begin with 8 molecules that have this functional group that is called tigleic acid. So these are tiglates. And then we had others. So if I'd let you smell them and ask you to categorize which ones are more similar, I'm not sure that you would be able to come up with this category because they're all kind of fruity, flowery, both these and those. Okay, but chemically, these share a large part of the molecule. So now you can ask, if a mouse is searching for 2 targets that are tiglates, does it matter if the background has tiglates or has non-tiglates? Okay. And let me walk you through this. So each square in this matrix is a specific combination of the number of tiglates and the number of non-tiglates. Okay, so this here is 2 tiglates, 3 non-tiglates, so a mixture of 5. Okay, and these are mice trying to detect tiglate targets. And the color codes performance. So yellow is good, blue is bad. Okay, and you can see that the color is organized kind of in columns, right? Going from left to right reduces performance. You go from yellow to blue. But if you go up and down, it doesn't do much. Okay, so for these mice detecting tiglate targets, the number of tiglates seem to matter a lot. The number of non-tiglates seems to not matter. Okay, so our intuition seems to work in this case. And I still think it's great that we can use our intuition in some cases. The only problem is, what if I now take a new bunch of molecules, maybe without tiglates? What do I do now? So this is nothing that we can generalize. So what we did, I skipped a lot of slides. I thought I'm going to have less time. So I'm going to try to explain this. And you stop me. There are a few steps on the way that we have no slides for them. Stop me if you don't understand. Yeah.
Maybe a website, but you already said it, but why do you have two targets? You said that it wasn't.
I didn't say it yet. I will in a second. So the other option is to forget about the chemical structures, forget about the molecules. I don't care about them. Really what I care about is the way the nose responds. So the idea is that if I have two molecules that activate the exact same receptor types, with the exact same affinities, with the exact same temporal patterns, then I don't care what they look like chemically, they will smell the same. Okay? That was the idea. So now how do you measure the responses of all the sensory neurons? We can't, but we can measure the responses of some sensory neurons, and the way we do this is image the glomeruli. So I showed you some glomerular imaging data before. Just see which glomeruli are activated on the dorsal surface. So we see typically about, a data set would have about 70 glomeruli that we could identify and responded to the 16 odors. And then you can come up with some variables that will look at the relationships between the glomerular activation patterns. Okay, so if I have a target odor and a background odor and they're really similar in their activation patterns, is this difficult? And for similarity, we looked at, there are different measures. It doesn't matter what we use. Let's look at the right side here. So you can take now all the trials that any mouse has ever done. This is 30-something thousand trials. And you can rate how similar the background activity is and the target activity. Okay, how similar are these two? And it goes from 0 to 1 basically, and you can see increased similarity, performance drops. So each dot here is, I forget now how many hundreds of trials. Okay, with this average similarity and this average performance. So more similar, performance drops a little bit, but this really is not extremely satisfying. You can already see we have something better. We then asked whether maybe you don't need to be similar, right? Maybe it's enough that you activate the right glomeruli to at least a certain level. So similarity is a really strict criterion. If glomerular A for the target, A was weakly activated and B was strongly activated, then for similarity you request the same thing. But what if we just blast both glomeruli, right? So this idea we called masking. And we had some way of quantifying masking. And we found that, yeah, if you just blast the right glomeruli, basically you're fine. Okay? You just need to mask the possibility of the odor being present. You don't need to have something really similar to it.
How do you mask it?
So masking is, so if I know that, I have a pattern that is the target odor, what I request for masking is that these glomeruli are activated to at least the level of the odor. Okay? If there are more, it's fine. I don't care. The relationship between them, I don't care. This would matter for similarity, but does not for masking. So it's basically if I give you a visual task, asking you, I don't know, trying to come up with, asking you if this pattern is the same as some other pattern that you've seen. Similarity would mean that in a different trial, I'll just give you a somewhat similar pattern, but still similar, somewhat similar but different. Masking just says, yeah, now I just do this, and you just you have no information, right?
And how do you get the measurements?
So it's all based on imaging the glomeruli, and then the way. So how do we quantify it mathematically? It's basically a ratio between the background and the target, and then bound between 0 and 1, assuming there's no more than one masking. So once you reach the target level, it doesn't matter what you do. Okay? And then averaged across all glomeruli, so you get one number. Okay. So this made us really happy because this is a psychophysical function, right? That fits some parameter of the stimulus to the performance of the animal. So now I can take, I'm going to have to do imaging experiments, but I can take any molecules and probably be able to predict the performance of an animal. Okay, so this is nice. So we have a behavior. We know something about the physiology. Now let's put them together. So it turns out that putting them together, all the really cool experiments that you can think of are really difficult experiments. So just before you do start doing them, is there some thought experiments that you should do, to think whether you're going in the right direction? So the first one we did was, so let's say I give you the glomerular patterns now, and just ask you to tell me whether the target odor is there or not. Okay, can you use some simple algorithm to read out the presence of the target from the glomerular inputs? And do you need feedback, some top-down modulation or something for that? And have you guys learned about linear classifiers at all? No. So what does a linear classifier do? It basically sums up all the inputs. So these are glomeruli, okay? Just sum them up, okay? But you don't sum them up just this simply. You learn how important is each glomerulus. Which glomerulus you should listen to more and which less, right? So you give them weights. So you multiply this. It's like an input vector, right? A list of glomerular inputs. It's one vector. You multiply each element by its weight, sum them up, and then if you can take this value that you found and compare it to some threshold and make a decision, that's a linear classifier. So really what the linear classifier needs to learn is just the best weights. What are the weights that will give you the best answer? Okay, so this is really a thought experiment, right? So we did that. Turns out that this linear classifier will perform as well as the mice. Okay, and this really alarmed me to begin with, because this tells you, okay, why come up with these complex models of top-down if it's so simple, right? You build a classifier. You can think of the classifier as a single neuron in pure form, okay? And he just solves the task.
The weights are according to the activation?
The weights are learned, but they depend on the activation, yeah. So large weights will be given to glomeruli that are activated differently between mixtures with the target and without the target. Okay. So, and this is something I was thinking about for a long time, and I ended up thinking that I don't think this is how it works. This is a kind of a network that you build for a very stable environment where you know all the target odors, you know all the background odors. So if you introduce a new background odor here, this fails. You need to know everything. You need to know their concentrations. You really need to know all the input patterns, and then it works. But I don't think mice have this option, right? So the question was, can we come up with some way of telling whether these attention ideas, right? Basically this feedback model claims that there's some sort of attention mechanism that tells the factory bulb, attend to this, so respond to it, but do not attend to that, so do not. So can we show something like that? And here now the two targets are important. So how do you show attention? What's really the hallmark of someone attending to something? Yes. More than what? Background. So more than two things. More than he would be if he was not attending, but also more than the background. So attending to something has a price. Has everyone here seen this movie with this clip with 6 people, 3 of them I think wearing white, 3 wearing black, passing the balls to each other? Has anyone not seen it? I'm going to ruin it for anyone who's not seen it. But anyhow, you're asked to just count the number of times that the ball is passed from a white shirt to a white shirt. And they walk around the room. And then in the middle of the clip, a gorilla comes into the room, stands in the middle of the room, does that, and then leaves. And typically people, when they try to count the passes, they don't see the gorilla, right? So this is the price. You were paying attention to something, you missed something else. So for me, this is the hallmark of attention, really. It's not the fact that you can do something, it's what you missed. And the same with the cocktail party problem. If you decided to listen to me, you're not hearing someone else, okay? So can we show something like that with the odors? That's why we need 2 targets, right? Because I want, typically, here's the dream experiment. I have the 2 targets, and I tell the mouse that on the next trial, you should attend to odor A, right? Because it's beneficial for you. Let's say whenever I tell you that, 90% of the time, you actually get odor A. So you attend to odor A, and you're very good at it. What happens if B comes? Can you still do the task, or did you miss it because you were attending to A? So these tasks you can do with mice, I think, but then you start training them for way longer. So the idea was to do something different, and that it's basically, instead of cueing the animal explicitly about what he should attend to, just use the fact that recent history is very strong at shifting our attention. Okay, so if I ask you now to do a visual task, maybe either here or here, and you get some random pattern of where it is, and then it follows. You get 4 times here. Now your attention is here. So basically, the idea was to do the same thing with the 2 odors. So we have this is a list of trials. 0 means no target odor is presented. These are the 2 target odors. So banana means a mixture with the banana, not just the banana. And strawberry is the other target odor. And then you get this random pattern, right? Half of them are going to be zeros, half are going to be with a target, and the targets are going to be 50% banana, 50% strawberry. Then I just take, only the target trials. So the zeros go away, and I get this sequence here, and I ask, is this banana more difficult than this banana? Because this one followed many strawberries, but this one followed a banana. Okay, so here, maybe he's attending to strawberry, he's going to miss the banana. Here, he's already attending somewhat to the banana.
But he gets the mouse gets the reward for both of them?
Yes, so importantly, the mouse gets the reward for both of them, and not only that, he needs to do the exact same action for both of them. So there's no getting used to pressing on one button and then pressing it accidentally. It's the same button. Okay? The other button is these trials. So I don't have it. So what we've done is we switched, instead of just one lick port, now they have 2. So they lick left or right. So left would be zeros, and either one of these is right. Now it's not 3 days anymore to train them. It's about a month. But yeah, so we just analyzed this, and what we found is that there's a very strong effect of the sequence. So I should have brought this up one by one. Just look here. So the x-axis here is how far in history we're looking back. Okay, so 0 means this is the current trial. Their performance on the trials with a target now is about 80. This is one mouse, so it's about 75%. Most mice have a bias towards the left. I'm not sure why. Maybe the drop is a little bit bigger there or something. Now, 1 means that you had the same thing as you had here, and 0 means that you had something different from what you had here. Okay? So going one trial in history, if you got the same target, then you're better. If you got a different target, then you're worse. And you can keep doing this tree, and at some point it becomes a mess, either because we have less trials of this specific sequence, or because their memory is not long enough for 3 trials in history. And this is same mouse, but now looking at the response times, not whether they're correct or not. And you can see that if you had the same target, you reduce your response time. Okay, that's another element of attention. If you were surprised by which target arrived, then you require longer response times. Okay, these are same but averaged across a few mice. So it seems that we in this task, which is basically the same task beyond the fact that now there are 2 lick ports, we can see hallmarks of attention, right? Signs of attention. So the idea of a linear classifier wouldn't do any of these. It doesn't care about these. So it looks like expecting something will affect your response, and this expectation presumably is not from the nose, so it needs to come up from, reach the olfactory bulb or cortex from somewhere higher.
Can you show again how can we see the expectation?
Yeah, so on this trial you had a target, either strawberry or banana. Okay, let's say it's a strawberry. This is a trial with that. This is splitting these trials. Okay, this is just all trials. Now I'm splitting them to the ones that also followed a strawberry or the ones that followed a banana. Okay? So you're sensitive not just to, your decision is not just based on what you receive now, but you're better if what you receive now is the same as what you received last, just the last trial. Okay? So I interpret this as the expectation. Yeah, and this one mouse already did 15,000 trials or so. It requires a lot of data to collect to believe these, because proportions are very sensitive to small n's. But anyhow, so we think we see signs of attention, and therefore it's worth looking at this model. I'm just going to, since time is up, I'm going to just show you where we're taking this now. So there are a few predictions of this model that I think are really worth looking at. First one is that the responses of mitral cells in the olfactory bulb during behavior, first of all, obviously depend on whether they receive feedback from cortex or not. So top-down inputs. So, and also we predict that this dependency will also depend on whether this mitral cell responds to a target odor or a background odor, right? If you respond to a background odor, you may be inhibited. If you respond to a target odor, you may actually be excited. So the idea is to record from mitral cells in the mice as they perform the task and silence the input from piriform cortex in a subset of trials. Right? So now if we can do this with optogenetics, maybe 20% of the trials, you turn on the light and you record this basically responses to the same mixtures during the same task, but just without the feedback. So the prediction here is that, okay, I already told you what the prediction is. Second thing that is, for me, really interesting is recording from piriform neurons. So not all piriform neurons project back to the olfactory bulb. The estimates are around 30 to 60%, but no one really knows. But if we can record from these neurons that project back to the bulb during the task, right, and identify them as projecting the bulb to the bulb, there's some scheme here to allow us to do this. Then there are very interesting questions regarding, first of all, when are they activated? Do they respond to the odor, or do they start responding just when you expect something, right? Kind of prepare the olfactory bulb to respond correctly when the odor comes. But also how they respond to target and background odors. So there is some stuff here. And the last one is really I think the strongest, I don't know if prediction, but the model kind of depends on the fact that there's some mapping between glomerular inputs and feedback from the cortex, right? If you want to correctly inhibit and excite the right neurons, and the mitral cells are mapped, right? They're based on the glomerular inputs. Then there needs to be some correspondence between the feedback map and the feed-forward map. And there's some experiments that we're planning to run using imaging to just image both axon populations when the mouse is performing the task. So basically, that's all I had to tell you. That's it.
היי, טוב, דבר ראשון זה ממש מעניין.
תודה.
כן. ויש לי שתי שאלות. אין הרי גידול מבחינת ריח. אז אנחנו מבינים, אנחנו מצליחים להבין את מקור ריח על ידי גידול של הראש.
גידול של איפה הריח למחר. לא יודעים שהם.
לא יודעים שהם? כאילו, לפחות לא רואים, אבל אין לנו עדות שיש.
אז עשו די מעט ניסויים. קודם כל יש שאלה ברמה ההתנהגותית. האם חיה כלשהי יודעת להגיד, תעשה לה פעם פה ופעם פה, והיא לא נמצאים אחרים, האם היא תדע להגיד מאחרים. הטענה היא שאחרונים הכל דעות צריכים לעשות. נורא קשה לעשות את זה באמת קונטרול, כי זה השאלה היחיד שהשתנה בניסויים. אבל הטענה כרגע זה כנראה מצליחים. אבל נכון שאתה יכול לצאת ראש,
גם לא יכולים, או שלא עשו ניסויים כאלה בבני אדם.
גם שם יש ניסוי אחד שאני יודע שיש טענה שהם מצליחים.
אוקיי, והראית את ה-trajectory כדי להגיע לזה, אז אנחנו לא רואים שהם הולכים בקו ישר, רואים שזה כל הזמן את השיפטים האלה.
נכון.
אז זה כאילו לאסוף יותר מידע.
יש שם, זה ברור שיש שם, אתה מאבד את הריח וחוזר אליו, ומאבד אותו וחוזר אליו.
כן.
אתה לא...
כן. אוקיי, והייתה לי עוד שאלה, סתם מקורס אחר, אני זוכרת שיש שני סוגים של גלומר אולי לכל רצפטור או משהו כזה. שניים, נכון? כאילו לא התייחסת לזה? אוקיי.
אני חושב שאמרתי במשפט אחד.
זהו, אבל רק רציתי...
מספר גלומר אולי הוא בערך כפול.
כפול, אוקיי.
אבל אין, הסיגניפיקנטיות שזה לא ברורה לאף אחד.
כן.
יש שם איזה סימטרי, אוקיי, אני לא תיארתי את זה, אבל יש מסכים על פקטור אחד, אז אתה קודם כל זה קו שסביבו זה מין סימטרי כזה.
אוקיי.
אז קוראים להם מירו, אבל לא ראיתי שמישהו הציע משהו על למה זה יותר טוב מכלומר, אחד יותר גדול.
לא, סתם חשבתי לעצמי שמה שכאילו שמתי זה התקשר למשהו. אתה קורא למה שאתה מוצא קשר, כי אתה אומר, אוקיי, זה היה הרבה פעמים על השמאל נגיד, ואז על הימין. אבל האם זה לא יכול להיות שיש לי מין איזה ממוצע כזה, ואז ברגע שעשית טוט, אז עלה לי הסתברות לטוט, ואז נגיד אני חושבת שזה כמו על פריור. אוקיי, נגיד יש לי פריור חזק לטוט, כי היה לי 4 פעמים טוט, אז הסיכוי שלי לטוט עולה על הבננה.
אני חושב שזה בדיוק ככה.
אז זה לא קשר.
כן, אני אגיד לך למה זה כן.
אוקיי. אולי אנחנו פשוט אומרים את זה במונחים שונים, אבל...
הפריור הזה גורם לך לחשוב שטוט הוא הצפוי.
נכון.
אם את משתמשת במידע הזה, איך את משתמשת במידע הזה?
מה זאת אומרת?
טוט הוא הצפוי מול בננה.
נכון. אז אני יותר אתן לטוט.
כן, אבל התגובה שלך לטוט בבננה היא אותה תגובה.
נכון.
אז זה לא משנה למה תיתי יותר. אם את רוצה, את תזהי גם את הבננה באותה מידה.
נכון.
אבל את לא מזהה את הבננה באותה מידה.
נכון, כי יש לי פריור.
אבל איך פריור זה משפיע?
נגיד, אני פשוט הייתי חושבת על זה שנגיד אני אעשה באמת כאילו באמת שנגיד יהיה הרבה יותר פעמים טוט מאשר בננה, ואז דווקא לשחק עם העניין של ה...
זה הסיבות.
כן?
אבל זה עדיין פריור.
נכון.
איך אתה משתמש עם הפריור? פריור שיגיד לך איך אתה אמור להגיב, היה מכניס אותי לבעיה. אבל פריור זה לא אומר איך אתה אמור להגיב, נכון?
נכון, אבל הוא עדיין משנה לך את התגובה שלך, כאילו את הזמן תגובה שלך, את הטעויות שלך, נכון?
איך זה משנה?
לא יודעת.
אז בעיניי attention זה התוצר הסופי. מה גורם לך attention דבר אחד? זה או הערך שלו או כמה הוא צפוי.
אוקיי.
אז אם יש לי שני דברים שהם בערך זהה, אבל אחד יותר צפוי, והם מתחרים, אני לא יכול להתמודד עם שני דברים, אז אני אקשיב לצפוי, כי שם הריוורד.
ואם אני עושה שנגיד הטוט יהיה לי הרבה יותר, אבל היה לי בננה עכשיו 3 פעמים, כאילו אני חושבת על ריסטיים כלעומת, כאילו פריור שהוא יותר קדום ופריור שהוא יותר...
זה עניין מסובך.
כן.
אבל... כאילו
אם עדיין אני אתן לבננה הזו שהייתה לי 3 פעמים, או שאני אתן לטוט שהיא יוצג יותר.
זו שאלה שונה. אז בוא נסכים לרגע שנקרא לזה פריור, ושהפריור, אתה משתמש בו לכוון את האטנשן שלו.
נכון.
עכשיו השאלה, מה משפיע על פריור? איך אתה מייצר פריור? זה מה שאת שואלת. איך המבנה של ההיסטוריה קובע את הפריור שלך הרגעי? דברים גם את הנביצ'טיין.
לא, אני פשוט טוענת שיש 2 פריורים, שזה פשוט תלוי...
זה נהיה מרחב גדול שאני אדע להתמודד איתו. אבל אולי אנשים אחרים יודעים להגיד משהו. אבל יונתן הנביצ'טיין זה היה החיים שלו למשך 10 שנים.
כן.
זה על היסטוריה של... או אולי עדיין.
תודה.